For decades, the unsung heroes of aging science have been mice, tiny, twitchy, crumb-sniffing mammals that have spent generations getting old in the name of our potential immortality. If you've ever read a headline like "Scientists Reverse Aging in the Brain," only to find it was in a 14-month-old mouse named Steve, you know the roller coaster of hope and confusion this field delivers. But something exciting is happening. After years of running geriatric obstacle courses in rodents, aging research is finally putting on human-sized pants and stepping into the real world. The lab mice may not be retiring yet, but they're no longer the only game in town. Let's start with the mouse obsession. Why mice? They're small, cheap, reproduce faster than your cousin's conspiracy theories, and their genetic and biological systems are surprisingly similar to ours. About 85% of their protein-coding genes match human ones. But perhaps more importantly, mice age fast. In 2 years, you can observe nearly every stage of aging: frailty, cognitive decline, muscle wasting, immune dysfunction, and hair loss. Poor little guys. That speed makes them ideal for studying interventions like calorie restriction, rapamycin, senolytics, or even NAD+ boosters without having to wait decades like you would with humans. It's like binge-watching aging instead of tuning in once a year. However, what works in mice doesn't always work in people. Mice don't get Alzheimer's or atherosclerosis in quite the same way we do. Their telomeres are wildly different in length, their metabolisms are faster, and frankly, they've never worried about their 401k or stood up too fast and pulled a hamstring. Many mouse studies use genetically identical animals raised in extremely controlled environments, essentially the opposite of human life, which includes stress, pizza, pollution, and TikTok. So while rodent research is invaluable for spotting promising interventions, the leap from "this mouse lived 20% longer" to "this grandma just ran a marathon at 92" is a big one. That's why the last decade has seen a seismic shift in aging science. More and more, researchers are pushing beyond the cage and running clinical trials in, you guessed it, actual people. Large-scale longitudinal studies, like the Dunedin study, have tracked human health for decades, helping to find new biomarkers of aging such as DNA methylation clocks and inflammaging profiles. These tools let us estimate biological age more accurately than candles on a cake, and they're opening doors for testing whether lifestyle changes or medications truly slow aging in humans, not just mice who've never eaten a donut. Trials like TAME, targeting aging with metformin, are testing whether a common diabetes drug can delay age-related diseases in non-diabetic humans. Senolytics, which aim to clear out zombie-like senescent cells, have already moved from mouse miracles to early human studies, some suggesting improved strength, less inflammation, even fewer falls in frail seniors. And supplements like omega-3s, vitamin D, and NAD+ precursors are being scrutinized for their effects not just on symptoms, but on aging markers like telomere length, mitochondrial function, and gene expression. But with great promise comes great complexity. Human trials take longer, cost more, and face the messiness of real life: diet, sleep, stress, genetics, the occasional midnight cheeseburger. Unlike mice, humans don't all live in the same lab and eat the same kibble. But this complexity also makes the results more meaningful. We're not just trying to extend life in a bubble, we're trying to make human lives longer, healthier, and more awesome, despite all the chaos. So where's this all heading? Toward a world where aging is treated like a modifiable condition, not an unchangeable fate. Thanks to epigenetic clocks, muscle scans, and blood-based biomarkers, we're developing the tools to test whether an intervention is working in months instead of decades. That means faster feedback, better personalization, and yes, more people living like 50 is the new 30 and 90 is the new 70, but with fewer hip replacements. In the end, mice gave us a roadmap. They showed us that aging isn't just wear and tear, it's driven by biochemical pathways that can be nudged, tweaked, and occasionally hijacked. But it's the humans, the volunteers, the patients, the stubbornly curious researchers, who are making the journey real. Aging science is growing up, stepping outside the lab, and asking not just how long we can live, but how well. And if one day we do crack the code of longevity, let's all agree to raise a glass to the humble lab mouse, preferably one filled with resveratrol, omega-3, and a vitamin D chaser.
Cheers to you, Steve.
You ran so we could walk briskly into our centennial birthdays.